So for the first speaker for this session will be Kirsten Meyer. Kirsten is a principal staff scientist in cellular genetics program at the Wellcome Sanger Institute, UK, working with Sarah Teichmann's group. She received her PhD from the MRC Laboratory of Molecular Biology, University of Cambridge. Her research focuses on characterizing human lung and airway atlases, also including those in disease contexts like asthma and COVID-19. With her recent exciting work on local and systemic responses to SARS coronavirus 2 infection in children and adults. Kirsten is also instrumental in assisting us in the establishment of the single-cell facility at Mahidol University, for which we are greatly thankful. Her talk's title today is Spatial Atlasing Reviews and Airway-Associated Immune Niche. So please welcome Kirsten.
Great. Waradon, thank you very much for those kind words. And thank you very much to the organizers of this conference for inviting me and giving me a chance to talk today about the work we've done on the adult lung. Now, just putting things into context a little bit, the lung is, of course, the primary gas exchange organ of the body, but therefore it also is a very important barrier organ. And of course, the lung is very relevant to human disease. There are many respiratory diseases, of which some of the most common are, for example, asthma and COPD. And just for COPD, for example, there were over 3.2 million deaths in 2019, including in low and middle income countries, really highlighting the relevance to health of studying the lung. And of course, with the recent COVID-19 pandemic, I don't think I need to remind anybody that respiratory infections are a very major health burden with many millions of deaths over the last 2 years. So in the context of all of that, it's really not surprising that the lung has been one of the organs that have been were one of the first ones to take up single-cell sequencing, and there are now many, many different publications that have single-cell RNA-seq data. And in fact, Lisa Sikkema and collaborators put out a very nice integrated lung cell atlas onto bioRxiv earlier this year, and that integrated lung cell atlas includes over 2 million cells from 444 donors and 46 studies, and indeed we also contributed a couple of studies to that effort. But importantly, all of those studies are primarily suspension cell studies. And so today I would like to tell you about our recent study where we're really trying to move from this suspension cell census to a spatial lung atlas. And in order to do this, we sampled the lung in a number of different locations, trachea, bronchi, as well as upper and lower parenchyma, to give the broad anatomical regions within the lung, but then we also took tissue samples and profiled them with Visium spatial transcriptomics to really begin to understand the much more fine-grained cell-cell interactions. And all of that, of course, will allow us to address the big question of what is the spatial context of the cell types and how does that affect cell function. This just gives you an overview of the experimental outline. We, in total, profiled about 190,000 cells, both from single cell and single nuclei, and Elo Madusun in our lab really led the cell type annotation. And I've shown here a UMAP that shows a very broad outline, but in total there were 80 different cell types that were annotated, and of those there were 11 novel cell types or cell states. In the interest of time, I'm not going to go through all of those, but in my talk we'll highlight just a couple. But together with this single cell data, we also generated the Visium data, as I already mentioned, and we were able to use the algorithm cell-to-location, which was developed by Vitaly Kleshtevnikov at the Sanger Institute. And this algorithm really allowed us to map the single cell data back onto the cell, or onto the Visium slides, where you have these barcoded voxels, and then you get a Z-score that tells you the abundance of specific cell states within that H&E section. And this slide really just shows is a proof of concept. Here we have a small airway, and we can see that both the ciliated and basal cells of the surface epithelium map to that inner lumen of this bronchus, just demonstrating that this works. And of course, we had multiple different sections, so we were also able to look at cross sections, which were manually annotated into different anatomical regions. And then here, for example, you can see that the multilayered epithelium indeed correlates with the secretory, the ciliated, and the basal cells, as you would expect. But of course, this also allows us to check which cells co-locate and allows us to see whether there are novel interactions that we might not have anticipated. But the spatial data is also really, really helpful in annotating cell types. So here I'm now showing the UMAP for the fibroblast compartment, in which we actually annotated several novel cell states, but I'm just picking out a single one, the peribronchial fibroblasts. And the reason I'm picking it out here is because when we use cell to location to map this to the tissue section, you see this very apparent staining just around the bronchus, which is in sharp contrast, for example, to the data we get with the adventitial fibroblasts, which map throughout the whole tissue section. And that immediately gives you some insight into what the cell function might be here, but it also really helps us in annotating cell types. So when we get two different cell clusters that may not have a very large number of differentially expressed genes, if we can see that there's distinct tissue locations of those cell types, that is very strong evidence that they really are different states. Just a little bit more on these peribronchial fibroblasts. As it turns out, we believe they're associated with COPD. And when we do a functional GWAS analysis, where we just look at the gene expression of GWAS-associated genes, we find those genes are overrepresented in the number of different cell types, including the peribronchial fibroblasts. And again, this is now also where we can harness the integrated lung cell atlas, because that includes mainly healthy, but also some diseased samples. So we used our annotations or our markers to annotate the peribronchial fibroblasts, and we're then able to ask within that population were the differences in gene expression between the controls and the COPD samples. And indeed, we see that many COPD GWAS genes are overexpressed, again, providing evidence that this cell type is associated with that disease. Another example of where the spatial location really gave us insight is the distribution of IgA plasma cells within the airways. And quite to our surprise, when we mapped IgA plasma cells to our Visium samples, they absolutely overlapped with the submucosal glands in the airways. Here's just a relatively large section, and then here are tracheal sections where you see very nicely that the gland cell type, the serous and the mucous cells, map to pretty much the same positions as the IgA plasma cells. But interesting, the mapping is quite different from B cells. And again, we can look at this across a number of different sections. And then again, here you see how in the glands, we have, co-location of the mucous duct as well as serous cells with the IgA. On the other hand, the IgG cells really mapped to what we called immune infiltrates and had very different spatial location. Now, actually, the knowledge that IgA maps to submucosal glands has actually been around since the 70s. But of course, now we have single-cell data, and we can really use this data to dig into the signaling circuits that might establish this niche. And we therefore carried out quite extensive cell-cell interaction analysis. And I'm really just showing one example here, where you can see that serous, mucous, and duct cells all express CCL28, which interacts with CCR10 that's preferentially found on IgA plasma cells. We did further analysis, and I come back to that later. Of course, we wanted to know whether this is all at the RNA level. Could we see the same effects at the protein level? And for this, we did IBEX staining, and this is work done by Nathan Ricoz from Menna Clatworthy's lab. And in the top row, you can see the epithelial cell staining really showing where the glands are located. And then underneath, you can see some IgD staining, but very strong IgA staining in blue, with almost a complete absence of IgG staining. And interestingly, we also see CD4 T cells in the bottom row that are quite abundant throughout these submucosal glands. And when we follow that up a little bit further, we actually see that these CD4 T cells map to regions that are high in HLA-DR. So these epithelial cells would not normally be expected to express HLA-DR, but when we stain, we see it's very clearly there, although it's not present in all glands, suggesting that maybe it gets expressed in response to stimulation or pathogens. And those regions that are high in HLA-DR, I can't use the pointer, I'm sorry, it keeps going, are also high in CD4 T cells, suggesting that the duct cells or the gland cells are actually able to act as antigen-presenting cells. So, given that I have a relatively short time to speak to you today, I'm not going to show you more about all the cell-cell interaction analysis that we did. Instead, I'm just going to summarize the results we obtained in this cartoon. So we were able to define transcriptionally a number of different cell types, such as the duct, the mucus, and the serous cells. We could demonstrate that they secrete CCL28 and CCL20, which recruit, in the case of CCL28, the IgA plasma cells, or CCL20, which can recruit naive B or memory cells, as well as T cells. And then we also found that the epithelial cells secrete a number of different markers that lead to the maturation and stimulation of IgA plasma cells. So, for example, APRIL can interact with BCMA TACI on the plasma cells to ensure longevity and maturation. Similarly, it can also interact with B cells to cause them to mature further. In addition, as I've already said, we know that MHC is expressed on some of the duct cells, likely leading to T cell activation, which can then provide T cell help. In other words, they're both T cell independent and T cell dependent mechanisms in action here. And this then leads to the formation of increased amount of IgA, which can be transcytosed into the lumen, where, of course, hopefully it can neutralize any viruses. So we believe through actually having done a very broad profiling, we've been able to put together a lot of different components of this immune niche and how it can increase the production of IgA for better immune protection. And we actually also earlier in the year published a relatively large COVID-19 data cohort, and in that we could show that indeed IgA plasma cells are enriched in COVID-19 patients, making the understanding of these pathways all the more relevant. And so my overall take-home message is really that having broad agnostic profiling together with spatial data can really help you to understand tissue function. And with that, I'd like to stop and just acknowledge everybody who did the work. This was all done as part of Sarah Teichmann's team at the Sanger Institute, but I'd like to particularly highlight Elo Madesoon and Amanda Oliver, who've contributed to this work, along with many other members of the lab and our Sanger pipelines. And our funders, again, were a number of different people, but in particular, I'd like to acknowledge Discover, the Wellcome Trust, and the CZI for supporting this work. Thank you.
Any question from the audience?
Hi, lovely talk. I was just wondering, so the signaling diagram you showed at the very end with CCL20, etc. How much of that have you thought about how to validate that? So I mean, I work in cell-to-cell communication space, and it's like everything's talking to everything. There's many messages. How do you actually prove that that's what's happening?
Yeah, I mean, that's a very good point, and it's very, very difficult. So we did try to look and actually obtain CCL28 knockout mice. But as it turns out, the mouse trachea are really very different to the human ones, and there are very, very few submucosal glands. And so we didn't actually see much IgA in the mouse trachea. So that makes it very difficult to validate in an animal model. So I guess at the end of the day, will be limited to demonstrating the cell-cell interactions, and we have actually demonstrated that a lot of these proteins are actually expressed in the right places, and that, for example, APRIL expression, CCL28 expression correlates with the amount of IgA present, but it's indirect.
Cool, thank you.
Great presentation, thank you. Maybe two-part, but one is related to what Alistair just asked, is how's the cell-to-cell communication analysis influenced by the resolution of spatial transcriptomics? Because obviously, if you have multiple cells, it may be harder to infer that.
Yeah.
And then the second question is, you profile different regions of the lung. I guess you have upper airways and lower airways. Do you observe similar pathways and mechanisms in which IgA is expressed?
Yeah. To the first question first, of course, we realize that these voxel of 50 microns and Visium are not single-cell resolution and have probably tens of cells in them, which does limit the resolution. But it's quite interesting, we also carried out NMF analysis of the Visium data. And then when we increase the factors, we actually see resolution that is much smaller than the 50 microns, I think largely because often a voxel will only hit a small section of a particular structure. And when we do that, for example, we can see that the basal and suprabasal cells of the epithelium map together, but map away from the ciliated and secretory cells. So actually, somehow the slightly random nature of the voxels, I think, actually helps you in terms of looking at the cell-cell co-occurrence. But obviously, if we had greater resolution, I'm sure we'd find more interesting things. And then the second question was the big differences. We do see some differences in gene expression across, for example, from upper to lower airways. For example, ACE2 expression differs, which is more highly expressed in the upper regions. But with the relatively few locations we've sampled, and with the actual samples not having been microdissected, it's actually quite difficult to get statistical significance.
Thank you.
Thank you, Kirsten.
Hi. It is my pleasure now to introduce our second speaker, Professor Muriam Choi. Professor Choi's expertise lies in functional interpretation of human genetic variants using genomics and bioinformatic methodologies. After doing his PhD in Duke University and then his postdoc at Yale, he joined the Seoul National University. And over there, his team studies the genetic mechanisms of rare disease pathogenesis with a focus on cases with neurodevelopmental defects. His lab has also established single-cell sequencing, and today that's what he's going to talk about. He's going to talk about genetic dissection of COVID-19 susceptibility. The floor is yours, Professor Choi.
Thank you. Thank you for a nice introduction, and also thank you for inviting me to this wonderful event. Again, I'd like to talk about COVID story, but I'd like to focus more on the equator and trying to see some difference from the previous studies. So we all know that for COVID, there are two major factors, age and comorbidity, that determines the severity of an individual. And of course, my question here, since I'm mostly from the genetics background, is whether there is any genetic factor contributing to this trait. So genetics of infectious disease has been traditionally very hard to study because of large contribution of non-genetic factors in the trait, and also involvement of many genes. But as you know, COVID-19 pandemic has provided an ideal cases for genetic study of infectious disease because because of the international focus and emphasis on the disease. And there has been, I think I could categorize the effort into two major ones. The one is more focused on the common variant approach, which is mostly done in GWAS. And there was another notable approach, which is on the rare variant association, and especially the Casanova group in the Rockefeller University has provided a list of genes that directly affect the severity of COVID susceptibility. So this just shows you that COVID HG initiative has made an interesting list of genes that contribute to the severity of disease. And also, Casanova group has published a number of papers, especially emphasizing the involvement of interferon signaling in the patient with a very severe type of COVID-19. So here, I would like to address several questions on top of it, so that are there additional variants that confer different severity to COVID-19 through gene expression changes, and genetically, how important are these EQTL variants? We are using the data set from Korean government and funded by Korean government. There has been a multi-omics data funded by Korean Center for Disease Control and Prevention. They have recruited 420 individuals with varying severity, especially mild and severe types. And they were equally contributed in terms of male and female, and also in terms of an age distribution. And they have many different types of omics data from them. EQTL, I don't think I need to spend time on that, but I'd like to emphasize that colocalization, people has been quite enthusiastic about the colocalization, but we all know that the success ratio is not that high, mostly 20 to 30%, and hopefully I can, although the time is not enough, but I'd like to show you why the rate is not that high. Also, we know that EQTL does not have a direct clinical implication, and of course, it needs tissue samples to assay. Especially to circumvent the minimal clinical implication issue, people has developed multiple different advanced EQTL strategies. For example, the response EQTL is also recently performed in our group using the NAFLD disease model, is trying to distinguish the group into different physiological status, so that you have a NAFLD group and non-NAFLD group, and GTEx as another control, and you can extract the EQTL signals that are specifically found in the patient group. And then you can start the story from there, that maybe you can make a hypothesis that this gene can be important in the pathogenesis of certain trait. And another advantage of that is obviously because of the genotype difference, you can differentiate the patient by genotype, so that you can make a different therapeutic approach depending on the genotype of the patient. Dynamic EQTL probably similar in terms of an idea, but they also look at the different time points or different, for example, different UMAP status and so on. Hopefully I can, at the end of my talk, I can persuade that GWAS gene, if you want to categorize the genes by their importance, GWAS gene and EQTL genes and response EQTL genes, they might differ in terms of a genetic significance. And I also would like to suggest that, although this is an ongoing project, I would like to suggest that EQTL and response EQTL genes can fill the individual level variations that are still left out by GWAS genes, especially for the complex trait like this. First, I'd like to start by acknowledging the student, my talented graduate student, Eun-young and Jung-ha, who has been tirelessly pushing this project. And also, I'd like to thank the Korean government for the fund for the generation of the data, and also for the sake of time, I'm not including all the network of clinicians and the data centers that made all different kind of multi-omics data. Okay, so the data is that are currently using is healthy individuals, influenza as another positive control, and also we have mild COVID-19 individuals and severe individuals, and they were sampled in 3 times during their treatment. And we also have data from whole genome sequencing and HLA sequencing and single-cell RNA sequencing of blood, PBMC, and also TCR, BCR sequencing data and serum cytokine level as well. We are working on to comprehensively call all the EQTLs from different kind of data, and collectively want to call them as a COVID EQTL. first to show you some of the single cell data. This is the obviously PBMC single cell clustering. We didn't, at this point, we didn't want to make too much sub clustering. So we have like an 8 clustering, major clusters, and we have called EQTLs. And for example, this is an EQTL that, so I'm sorry, so you are looking at 6 windows, and the 3 at the top is on mild individuals, and the bottom 3 are severe individuals. And obviously, column 1 to 3 are 3 different time points. And you can see that among those 6, this particular SNP shows quite an interesting association with a gene called S100P, and the expression is assayed in monocyte. And these all different window shows certain correlation, and which is also confirmed in the GTEx data as well. So this can be a good example of an EQTL. And response EQTL, for example, here is the SCAMP1 in monocyte. You can see that although all different 5 windows, there's not that much significant association. You can see that there's a special specific signal in the mild 1 time point. So this can be a good example of response EQTLs. So we first wanted to know whether this EQTL and response EQTL show different genetic signature from GTEx, from GWAS SNPs and among those as well. So first we looked at the distance from TSS and the EQTL position. And we have categorized 3 different groups, the EQTL in all groups, the one that I showed you right before. And EQTL, and you can also have an EQTL that showed in at least 1 group out of the 6 windows. And among those, you can also pull out the EQTL that are response EQTL that show the specific difference between the mild type and the severe type. And if you look at those, the distribution of their EQTL position from the transcription start site, you can see that the enrichment gets lower and lower. Also, we looked at the gene ontology enrichment. For the sake of time, just giving you one example, adaptive immune response enrichment gets much weaker as you go to response EQTL. And epigenetic signature, we relied on the 2 different databases, RegulomDB and HaploReg. And what we saw was that if you compare all SNPs in the genome and all eSNPs, and also EQTLs in all groups, and EQTL that we saw from at least 1 EQTL, 1 group, and finally response EQTL. And what you can see is that if you go from all SNP to EQTL in all groups, it gets stronger because the dark signal is the more strong category, functional categories. Whereas if you go from EQTL to all group to more specific response EQTL, it gets lower and lower in terms of a signature, which is the pattern is also consistent in different HaploReg databases as well. And I'd like to show you the notable COVID EQTL examples. So this is trying to correlate between the genotype of a whole genome and the serum cytokine levels. And what we were able to see was that interleukin-6, LRG1, and PTX3, they showed an interesting signature in certain specific window of the individuals. And we also tried to link between the TCR repertoire data with the genotype. And from the TCR sequencing data, we pulled out 4 different readouts of TCR distribution. So as you see here. And one example I'd like to show you today is one of the signals in STEMA3A. There were multiple signals found in that big LD block, which is 85 kilobase long. And this shows you that if you go to, this is EQTL that shows the same pattern among all different 6 windows, that as you go to the reference to non-reference allele, you have a much higher percentage of, higher number of expanded clonotypes. So what does that mean? So we have been looking into it, and STEMA3A is, as you know, might know, is a negative regulator of T-cell immunity. And there has been a small-scale GWAS on Chinese, and there was also a significant association with that LD block with the COVID-19. So linking some of the functionality between the EQTL shown here and the COVID-19. The final piece of data that I'd like to share with you today is that that I have been telling you that although EQTL is probably not as significant as EQTL or GWAS expression, GWAS SNPs. But if you look into certain pathway, especially in this case, is a very well-known COVID-19 infection pathway, you can highlight the genes with a yellow, which is an already known causal genes that are defined by Casanova's studies. When in addition to that, what we were able to do was we were additionally highlight the genes that were, although they were not directly causal, but they were interesting enough to regulate the expression of the certain gene by different condition and different genotype. So there were 2 genes that are involved in that process. And another pathway, if you're looking at, is a mitochondrial response to COVID-19. And again, there's a yellow genes in the very important genes. And we are also seeing that there are other response EQTL genes that may fill the gap between the major COVID-19 causal gene and that the gene may regulate the individual variation. So to summarize my talk, COVID-19 has provided an opportunity to study genetic contribution of infectious disease. We call the list of EQTLs and response EQTL that may confer individual level variation to COVID-19 infection. And we think that several COVID EQTLs are working in known COVID-19 pathways. And again, we are still working on trying to finishing up the project, and the complete analysis of COVID EQTL may help understand the mechanism of individual level variation. So this is my current working model. I'm trying to collect more data trying to fill this in. What I'm thinking is that not only COVID, if you have a very complex trait, there must be core genes that will cause disease when they are altered. And there are also GWAS or DEG genes that may contribute to the susceptibility to the disease. And I'd like to propose that EQTL or response EQTL may confer individual variability on top of it to trying to get a good profile of a certain disease. Thank you very much.
So we only have time for one question, if there's any.
Thank you. Very nice talk. In one of the slides that you're showing, in one single pathway, you have the GWAS gene over there, the yellow gene, and then the blue genes. And do you have any ways of systematically testing for that?
Right. That's a good question.
Yeah.
That's a good question, and obviously this is the way that we should go. So what we are thinking is then trying to, because what EQTL can give you is the individual level genotype information and what it can do to the individual. So hopefully we can finish up the profiling the individual pathway, and then we will get, we don't have a specific clue now, but we are thinking about statistically modeling them and trying to see how much of the contribution that these blue genes to do in terms of our overall pathway.
Thank you.
Thank you so much, Miriam.
Yeah.
The next speaker is Hamish King. So Hamish is a laboratory head at the Walter and Eliza Hall Institute in Australia. He received his PhD in molecular epigenetics at the University of Oxford before he became a Sir Henry Wellcome postdoctoral fellow in Louisa James' lab at the Blissett Institute, Queen Mary University of London. In close collaboration with Sarah Teichmann at Sankey Institute and William Greenleaf at Stanford, with highly notable works in characterizing human B cells and germinal centers. His current research aims to dissect molecular mechanisms that underpin global and locus-specific epigenetic regulation of gene expression in human B cell maturation using single-cell and epigenetic approaches. Today, he's going to talk about single-cell genomics to map immune cell state and predict cellular etiology of autoimmune response. Welcome, Hamid.
Thanks, Waradon, and look at him go back and forth across the stage, showing some dynamic chairing. Thank you very much, Waradon, and the organisers for inviting me to speak today. It's an absolute thrill to be here and engage with the HCA community in Asia and beyond. And the work that I'll be sharing with you today was mostly done at the Bliss Institute in London. With this building here called the Neuron Pod was described by the Guardian newspaper as being an intergalactic porcupine. I've recently moved my lab to the Weehai in Melbourne, where we've got some very different spiny creatures running around the place. But really what my lab at Weehai is interested in doing is filling in the bit between the middle here. So we've known for a very long time, we're getting more and more information about the phenotype of different cells and different parts of the body, and we're accumulating more and more information about human genetic variation. And obviously I'm loving the theme of diversity at this seminar today. But this bit in the middle is really important too. How do we translate the genetic findings with changes in phenotype, and ultimately how does that link to disease and intervening in that? And my background's in gene regulation and chromatin biology, so the epigenetic mechanisms of this control are really interesting to me. But like a lot of questions in biology, the dynamics and context are essential. And that's the main theme of my talk today, where I'll try and convince you that we need to be asking these questions about gene regulation and dysregulation in the right place at the right time. And my system of interest is human B cells and their maturation in the germinal center. And I won't go through this in any great detail other than to say that in response to an infection, and naive B cells move from the peripheral blood into lymphoid tissues like the tonsils, spleen, and lymph nodes, whereby they enter this germinal center reaction, rapidly cycling back and forth between different states, acquiring affinity to an antigen, and then undergoing a differentiation decision to form memory B cells or plasmablasts or plasma cells. This is obviously very dynamic and hard to study in human tissue, but it's really important that we understand this process because defects can either lead to immunodeficiencies or autoimmune diseases such as multiple sclerosis, Kawasaki's, rheumatoid arthritis. And the really important thing is that as well as only being found in tissue and not in blood, these germinal center states are really strongly associated with age. As we know, most immune activity is. And this is work done from a collaborator, Lisa Varga, at UCI in the States, where she looked in tonsils from different aged children and adults, and we can see a very clear correlation or loss of representation of these cells associated with age from under 5 years old to adults. And this is really key because a lot of the genomic and including single-cell genomic resources of studying the immune system have focused on blood or on lymphoid tissues from aged individuals 50 years or over, where we know that we don't get a good representation of these cellular states. So to go about this, our study design was very simple. We took pediatric tonsillectomy samples from 3 to 7-year-olds and performed 2 studies. One was using single-cell RNA sequencing and VDJ to understand the questions about their antibody repertoires, and a secondary resource generating single-cell attack sequencing. I won't present the full study to you today, but you can go and access this data online at tonsillimmune.org, which included our study looking at gene expression and antibody patterns. What I'll spend a little bit more time talking today is the work we did with Will Greenleaf on the single-cell attack sequencing resource from the human tonsil, profiling just over 33,000 cells and 16 different cell states. And as an example of I think the power of single-cell attack sequencing or attack sequencing in general, I just wanted to show one example locus of how we can use this to understand questions about gene expression and regulation. So on the left here, this is the gene XBP1, which is a transcription factor important for regulating different immune cell populations. And on the left, I'm showing the attack-seq signal across the different clusters, and on the right, the RNA-seq expression. And you can see different patterns of accessibility. So although the gene promoter is accessible across all of these immune cell types, there's highly B-cell specific accessibility at this region here, highly T-cell specific at this region here, and highly plasmablast specific expression here. And when we compare that with the gene expression from the RNA data set, we can sort of start to make some inferences about the relative importance or potential functionality of these different regions. which can be really useful. So to change tack slightly, I'm going to talk a little bit about non-coding genetic variation in autoimmunity and how we can try and learn more about the potential mechanisms. So I mentioned before that defective germinal centers are a feature of many different autoimmune diseases, as shown by autoantibody production that have signatures of a germinal center maturation, and the formation of ectopic lymphoid germinal center structures in tissues they're not supposed to be in. And as has been touched on already a few times today, this genetic variation is very complex, polygenic, with many thousands of GWAS variants identified, but a major challenge being that they're often found in non-coding regions of the genome, where the obvious gene target is not always clear. So the approach that we took was to use epigenomic information combined with gene expression to try and predict and identify the cell states that it might be most important. So in this very cartoonish example, in cell type 1, we can see that the SNP exists in an open chromatin state, and the gene nearby, gene 2, is highly expressed. Whereas in cell type 2, it's occluded, it's inaccessible, and there's no gene expression nearby. So we would perhaps naively predict that cell type 1 is going to be the more likely penetrant population of cells that could be affected. So we took a resource of nearly 13,000 statistically fine-mapped GWAS variants from 21 different autoimmune traits, and then combined this with our single-cell RNA sequencing and attack data of the human tonsil. And just like to credit two of my co-first authors on this paper, Zohar and Kristen, who are from the Greenleaf Lab at Stanford. They were fantastic collaborators on this work, and they deserve a lot of credit as well. So we didn't just limit it to the tonsil, though. Although we thought it was really important to look within the tissue itself, we wanted to see the full breadth of the adaptive immune system, and so integrated it with peripheral blood and bone marrow data sets that have been previously published, and identified 37 different clusters of population across the 2 different assays. This gave us just over 200,000 regions of the genome that were accessible in at least one cell type. And when we intersected those cell type specific peaks with autoimmune GWAS variants, we found, like many others have found, that most of these SNPs are highly cell type specific, found in fewer than 5 cell types in the immune system. And reassuringly for us, that about a third of those were only found in cell types that were enriched within the tonsil, not the blood or the bone marrow. So we'll look now at some of those SNPs themselves. So here is a heat map of 460 of those autoimmune SNPs, and just showing their chromatin accessibility from the single cell attack data in the different cell types. We can also look at the linked gene expression as well, and the major take-home that I'd like you to get from this is highlighted here in these yellow boxes, that there were 2 distinct, really clear blocks of signal, if you like, that came out of both assays, the accessibility and the gene expression, and that these blocks of activity were really specific to the germinal center associated populations, both the B cells and the T follicular helper cells that are required as well. And to drill the point home, these are only found in the tonsil tissue, in the lymphoid tissue, not in the blood or the bone marrow. So it was essential that we had that tissue profiling in our study. I've got time to just show you one example of a locus where we identified some of these autoimmune GWAS variants. And it's a HHEX locus, which is a transcription factor recently described to be required for differentiation of memory B cells. So here there are 3 different SNPs, and these are associated with multiple sclerosis. And we had identified significant correlations between the accessibility of these SNPs and the regions that they're in, and the expression of this nearby gene. And just zooming in on these, shown here is the accessibility in germinal center B cells. We can see that it's, in this case, highly specific across several different B cell clusters for this SNP on the right, whereas this SNP here in the middle was only really accessible in the germinal center, and not very much so as well. So there's questions about how functional this is, but I think it's still a significant signal. And finally, looking at the expression of the RNA, we can also start to make some predictions as well. One of the exciting things that we found was that for one of these variants in particular, it was predicted to disrupt a KLF motif sequence, providing a potential mechanism. Although there are lots of questions with these type of analyses about which exact transcription factor KLF is expressed in those cells, is it actually binding that region of the genome? And these are hard questions to answer on scale across the entire genome. Of note, we found many other genes associated with the germinal center were predicted targets as well from this study, including the master regulators BCL6, IL-21, IL-21 receptor, and so on. Some of these are known, but some of these, obviously, we found many that have unknown functions, and they're the ones that we're really excited about as well. So unfortunately, from our study, we can't predict whether these variants are likely to be a gain of function or a loss of function, because we're profiling healthy individuals who don't have these variants. And the existing QTL resources don't profile these cells. So that's something that we're keen to generate going forward if people are interested in having that discussion. But for the moment, we're left with a model of dysfunctional germinal center regulation that either an upregulation or downregulation could actually drive autoimmunity. And that's if we had a function, a variant regulating a germinal center associated gene that might regulate differentiation, proliferation, or survival. Either an up or down regulation of these processes would result in an altered homeostasis, disrupting the selection required to prevent autoantibodies, more potentially disrupting differentiation cues and pathways. So I'm now recently started at the WeHi in Melbourne, and sort of taking on from this study, I've got lots of fun projects and exciting work trying to understand how this dysregulation occurs, both at the enhancer-promoter interaction level, so pursuing a lot of these targets we identified using single-cell functional genomics like CROP-seq, CRISPR-I and CRISPR-A, and prime editing, or more genome-wide questions about how genes are controlled. We've also got collaborations working on immunodeficiency, including human vaccine trials in some of those rare cohorts, and we use a range of experimental computational work. If you're interested in any of that or just like to chat about science, do get in touch, and very happy to meet more of you in the coming 24, 48 hours. With that, I'd like to say a big thank you to my current team at the WeHi, who are great helping things get set up, but a big thank you in particular to Louisa James at Queen Mary of London, who is my postdoc supervisor, a clinical collaborator at the Royal London, Nara Orban, Sarah and other colleagues at the Sanger Institute, Will and his team at Stanford, and finally to all the patients and families who donated their tissue. And with that, I'd like to take questions. Thank you.
So because we're really running behind the schedule, so maybe one short question. Thank you. Apologize for that.
Hi, I'm Kaz from Riken. I'm also studying autoimmune disease genetic biology. Oh, nice talk. So a quick question. So you showed a nice example that fine-mapped GWAS SNPs co-localized with ATAC-peak, and you say that it is quite difficult to test allelic effect on that ATAC-peak, but have you checked the allelic specific effect on the ATAC-peak? Maybe even if limited number of sample, you could you might have some heterozygous individuals who have heterozygous SNPs.
Yeah, definitely. We have not really looked, to be honest. I think we're not really expecting to find them, so we didn't spend the time sort of going through the analysis, but it's something that we hope to do as we've now got a lot more data sets as well from this tissue, including the multi-omic assays. So I think as we get more patients as well, that's definitely worth doing, but we've not done it yet. It's a good point. Yeah.
Thank you so much, Hamish. So the next speaker is Regina Hu. Regina is a postdoctoral associate in Rosa Vento Thomas Lab at Wellcome Sanger Institute. She received her PhD in microbiology and immunology from University of Singapore before taking her first postdoctoral fellow position at Nanyang Technological University in Singapore as well, prior to her current postdoc position. In the Vento Thomas Lab, she has led, designed, and established the first comprehensive multilineage single-cell map of human placental tissue ex vivo and trophoblast organoid models. Today, she's going to talk about a single-cell multilineage map of infection during early human placentation reveals unique responses to pathogens. Welcome, Regina.
Thank you, Waradon, for the kind introduction. So first of all, I'd like to thank the organizer for inviting me to give a talk on our unpublished work on single-cell multilineage map of early placental infection. So the placenta itself, it's a very important organ during pregnancy, yet it is also one of the most poorly understood organ. So this nice electron, this nice electron micrograph here shows an infected red blood cell with the parasite malaria that is latching onto the epithelial cell of the placenta. Now, disease such as this is highly prevalent in low to middle income countries, particularly in developing countries, but yet the insights onto the early onset of the infection that happens at the placenta, particularly in the human placenta, is not really known. So this is because there are a few key challenges in these aspects, because first there is limited access to fresh placenta tissue per se, either from healthy or diseased donors. And then there is also important to note that there is also key differences of placenta between the animal and the human itself. So particularly this is true for early pregnancy. So we believe that there is a need in the case to develop a proper and better placenta in vitro model. And in our team, we embarked on a journey to develop a multilineage placenta in vitro model to better understand how infection occur during early pregnancy. So the cartoon here shows the villus tree, which is the main structure of the placenta. So it comprises of multilineage. So it comprises of multilineage cell type that made up of fibroblasts, macrophage, endothelial cells, and as well as trophoblast cells. So what you can see here, trophoblast is actually the unique epithelial cell type of the human placenta. And throughout my presentation later, I will particularly highlight trophoblast cell types. So the placenta plays an important function during pregnancy. So I just highlight the four main key functions of the placenta. It first protects the fetus against harmful external materials, and it also ensures proper nutrient and gaseous exchange into the fetus. And the placenta itself is also a main endocrine organ that promotes the production of pregnancy hormone. So all of these functions has to operate in an optimal manner to guarantee pregnancy success. So now if you look at the villus tree itself, its microenvironment is very unique because it is directly exposed to the space known as intervillus space. So this space is actually directly exposed to maternal blood where infections can potentially occur. So we are particularly interested in looking at the blood-borne infection coming from the maternal side. And with this unique tissue microenvironment, we ask the question, how do early infections actually affect the overall function of the placenta? Particularly, how does this different cell type lineage actually respond to infection? So to answer to these questions, we developed a novel multilineage placenta in vitro model, and then we perturb them with three major classes of pathogens, which I will describe later. Then we also profile the different lineage with single cell and single nuclei multi-omics. And then we also finally validate our data set, particularly on the trophoblast lineage in the trophoblast organoid system. Due to time interest that I had for my talk, I will only be focusing on the first 2 points. So to develop a multilineage placenta in vitro model, we use a three-dimensional tissue piece from a first trimester pregnancy termination placenta tissue, from surgical terminations, from healthy surgical terminations to be specific. So basically we embed the tissue ex vivo on a transwell culture system, and then we actually took a lot of effort to re-optimize the culture media from our collaborator, Toko et al., that will enable the tissue to survive in this microenvironment. And also we re-optimize the media specific for each of the pathogens that we perturb into our model. And this tissue is also co-cultured on a specific percentage of extracellular layer matrix that that is critical to ensure tissue viability and infectability at least 72 hours post-embedation on this system. So with that, we perturb our model with 3 major classes of pathogen with very clear distinct mode of action. I will not go into detail on the pathogens itself, but I would just like to highlight that we actually we managed to infect our model with Plasmodium falciparum, which is a very classical infection in the placenta that binds. Basically, it just binds onto the placenta tissue itself. And then we also included 2 pathogens, Listeria monocytogenes and Toxoplasma gondii, which is known to actually cross the placenta and cause intrauterine infection in the fetus. And then we profile them on our infection system. We profile them, as mentioned, using single cell and single nuclear RNA sequencing between infected and our uninfected match control of the same donor. And basically, we want to characterize how all the different lineage responds to infection across all the 3 pathogens. So what you see here is a UMap of integrated data sets from all the infections that we have done. So basically, we are able to recapitulate the major cell types of the human placenta, namely the trophoblast, macrophage, endothelia, and fibroblast cell type. So this cell types was basically annotated based on marker annotation, and we also project this UMap to our unpublished maternal-fetal interface data set through logistic regression and label transfer learning, so that we are confident that each of the clusters are a true particular cell type that we want to see. I would also like to highlight that here, what you see is that the outermost layer of the placenta tissue is also is considered multinucleated. So it's known as multinucleated syncytia, or in short, syncytia trophoblast. So with that, we also embark to profile single nuclei RNA-seq. This is important for us to capture most of the syncytia nuclei that is coming from the syncytia trophoblast. So what you see here is actually a small subset of our single nuclei data sets. So with the interest of time, I will only be focusing on the trophoblast cell type here, specifically only focusing on Plasmodium falciparum infection. So this IFA figure here shows that in our model, this is the ex vivo tissue model, where the parasite is actually binding onto the syncytia trophoblast. So I'm just going to give you a brief overview also, because in the next couple of slides, I will be only focusing on the first 3 sublineages of the trophoblast. So, for example, what is syncytia trophoblast actually arises. Syncytia trophoblast actually arises from the another progenitor trophoblast cell types known as villus cytotrophoblast, or in short VCT, which fuse to become VCT fusing, and then finally into syncytia trophoblast. So what happens to the syncytia trophoblast upon malaria infection? So we did a differential gene expression analysis and this gene ontology enrichment analysis as well, to enable us to see what are the key features that are different upon malaria infection, particularly at the outermost epithelial layer. So on first hand, you can see I only show a specific gene categories that we think is very interesting. So on first hand, you can actually clearly see there is a significant a change or downregulation in genes that are involved in the core function of the placenta. So for example, genes that are involved in the hormone biosynthetic process pathway, genes that are involved in nutrient uptake and the growth factor of the fetus is all actually downregulated upon infection with malaria. On the other hand, you see genes that are genes program that is upregulated involved in protein catabolism. We also see the similar feature being transduced into the neighboring cell type, which is the BCT fusing and BCT, as I mentioned, suggesting that there is like a systemic effect happening, despite the fact that the parasite does not really infect the neighboring cell type. So it's more like a bystander effect that we can see in our model. On On the other hand, also we see a very classical upregulation of pro-inflammatory cytokines and chemokines, particularly CCL3 and I1 beta for on the syncytial trophoblasts as well. And we also see the similar feature in the neighboring cell type that is not infected by the malaria parasite. So this upregulation of inflammatory cytokines and chemokines tell us that our model can also be induced in terms of, this is known, so our model can be induced by the infection as well. We also correlate whatever we see in the transcriptomic to the whole culture supernatant profiling. So we use Luminex profiling assay. Basically, it profiles the protein level of the cytokine in the culture supernatant, and we can see a good correlation in terms of that. However, I would like to highlight that this is only the subset of the trophoblast data set. So whatever we see from the culture supernatant, it slightly also coming from the fibroblast and the macrophage lineage that we have in our multi-lineage model. So in a very quick short summary, I've shown that in our model of infection, using malaria as an example, we see a very strong induction of pro-inflammatory cytokines and chemokines. This is known particularly for malaria parasite infection. But what really intrigued us is that according to this, we also saw dysregulation in the core placenta functionalities. So you see clearly downregulation of the genes that are involved in all these key important placenta function, suggesting that this tissue balance and homeostasis actually is very important in terms of maintaining the overall integrity of the placenta, the function of the placenta. And this balance of such will likely lead to disease pathologies. So because I was told that my talk has to be very short and concise, so I will just highlighted a very simplistic example on how the trophoblast from the placenta responds to one of the key pathogens, Plasmodium, using a model that we have developed, and also using single-cell RNA-seq to understand how the subtype of the trophoblast actually responds to the infection. So right now, ongoing is that we are building a multi-lineage map of all the lineage that we have in our model, and we also correlate it with the two key pathogens that I mentioned, namely Listeria and Toxoplasma, which clearly has very different mode of action. So they are actually the one that actually go into the placenta. So we are trying to understand how they are similar or different across different lineage and across different pathogens. And this will give us then a working hypothesis on how actually early infection cause inflammation and breakdown in tissue homeostasis, and at the end affect tissue function. So with that, I would like to thank the members from the Bento team, and also this project is co-led with our PhD student, Elias, and also our collaborators from the Sungnak Institute and University of Cambridge. And finally, I would like to thank all the donors from the Human Developmental Biology Resource, UK, for all the placenta tissue, otherwise this would not be possible. Thank you.
Thank you so much, Regina. So because we're really running behind the schedule, so we need to move to the next speaker. Thank you so much, Regina. So the next speaker, who is the last speaker for the session, is Rilya Edahiro. Rilya is a graduate student at the Department of Statistical Genetics, Osaka University Graduate School of Medicine, as well as a respiratory physician at Osaka University. He is currently working with Yukinori Okada, studying GWAS and post-GWAS analysis of Japan COVID-19 task force, along with a contribution to COVID-19 host genetic initiative. Today he's going to talk about large-scale single-cell RNA sequencing in Japanese, identify biological and host genetic involvement of innate immune cells in COVID-19 severity. Welcome, Rilya.
Thank you for a kind introduction. Today, I'd like to talk about COVID-19 multi-omics single-cell analysis in Japanese. As background, multiple studies have highlighted the dysregulation of immune response in COVID-19 using single-cell RNA-seq analysis. However, the immune response of the host to SARS-CoV-2 still remains unclear. In addition, COVID-19 GBAS, highlighted as an achievement of COVID-19 HGI, have demonstrated the influence of human genetic background on the pathogenesis of COVID-19. However, functional analysis mostly focused on the variant on chromosome 3, which have the strongest severity association in European, but confer a rare frequency in East Asian, suggesting the importance of additional study in non-Europeans. This is the overview of our study. We analyzed single-cell RNA-seq analysis and single-cell T-cell B-cell analysis from 73 COVID-19 patients and 75 healthy control of Japanese with host genetics data. This is a UMAP embedding of all PBM sheet. The total cell count is about 1 million, and first we focus on the monocytes and dendritic cell DC clusters. We performed a second round of clustering, then subclusters monocyte into 5 subsets and DC into 2 subsets. To reveal the compositional change between COVID-19 and healthy control in this cluster, we performed differential abundance analysis. First, we defined the neighbor, then fit the negative binomial model to the count of each neighbor using Milo. This analysis revealed that the proportion of non-clustered monocyte and PDC decreased in COVID-19 compared to healthy control. We also evaluated the differential abundance with severity of COVID-19, then found that there was a trend toward decrease in non-clustered monocyte and PDC in severe compared to moderate group. To analyze how cell transition differ in clinical status, we performed RNA velocity analysis using SCVero. The transition potential from intermediate monocyte to non-clustered monocyte was clearly observed in healthy. However, such a transition was not observed in COVID-19 patients. The cell transition from classical monocyte to non-classical monocyte through intermediate monocyte was previously found via in vivo experiment. These data suggested that the decrease of non-classical monocyte as a consequence of the downregulation of the cell transitions from classical monocyte to non-classical monocyte. We conducted a differential expression DE analysis between a moderate versus severe group in non-classical monocyte. PLD4, which digests single-strand RNA and single-strand DNA, was mostly downregulated in severe COVID-19. In addition, CXCL10, which is critical in response to various infectious pathogens, was also prominently decreased in severe COVID-19. CXCL10 is an interferon gamma-induced gene and exerts its biological function by binding to CXCL3. Therefore, we explored the intracellular interaction center of CXCL10 in non-classical monocyte. Cell-cell interaction analysis revealed that the activated T cell and NK cell have a strong interaction of interferon gamma receptor with non-classical monocyte as a sender, which was enhanced as the severity of COVID-19 increased. PDC have an interaction of CXCL10, CXCL3 with non-classical monocyte in moderate COVID-19, which was not observed in healthy or severe COVID-19. Cell-cell interaction analysis inferred the possibility that the dysfunction of non-classical monocyte could be one of the factors responsible for driving COVID-19 severity. We also performed BCR analysis at the single-cell resolution. We defined 4 clusters in B cells, and this UMAP is the UMAP embedding of the B cell colored by the clonal size from 0 to 50. Clonal expansion was most evident in plasmablast, followed by activated B cells. Right figures are the network plots of clone type. for disease status and per sample. In this network plots, chronotype with clone size over 10 was selected. Large clonal expansion was only observed in COVID-19, and very few chronotypes were shared among each sample. To illustrate the genome-wide hostogen risk of COVID-19 and identify disease-relevant cell types, we integrate information from our single-cell data and COVID-19 GBAS of 3 phenotypes. At first, we define the top 100 putable disease gene of 3 phenotypes using magma, respectively. Then we calculated the disease score of each cell at our single-cell data, and also performed the cell type level analysis, such as association and heterogeneity using SCDRS. These are the UMAP embedding of PBM sheet colored by disease score. Disease scores are similar across cell type when using self-reported GBAS, while cells annotated as monocyte and DC have a higher disease score than the other cell type when using hospitalized and very severe GBASs. We also assess the association between major 6 cell type and 3 phenotypes. There is no cell type enrichment in self-reported GBAS. However, monocyte and DC have an association with severe phenotypes GBASs. In addition, 3 cell types have a heterogeneity in association with severe GBASs. We investigated the monocyte and DC cluster in more detail. All of the 5 subsets have an association with severe GBASs, which was more prominent in very severe one. This data suggests that polygenic risk involved in COVID-19 severity enriched in innate immune cells. Finally, to gain a better understanding of transcriptional viability and dynamics regulated by GBAS-identified COVID-19 risk variant, we examined EQT effect separately for COVID-19 and healthy. We select 5 variants which were replicated in Japanese COVID-19 GBAS. First, we performed EQT analysis for major 6 cell types. COVID-19 risk variant shows different cell type distribution with significant EQT effect, suggesting context-specific EQT effect. Monocyte of COVID-19 have a significant EQ effect in multiple variants. Previous analysis demonstrated that monocyte is involved in the COVID-19 severity. We focus on this subset. The risk variant with EQT effect in monocyte of COVID-19 have a EQT effect specifically in classical monocyte, and no such EQT effect of IFNA2 variant were observed in healthy. IFNA2 has a very important role in type 1 interferon signaling pathway. Then we focus on this variant. These are the box plot of the EQT effect of the IFNA2 variant. The risk area T have a COVID-19 context-specific increasing dosage effect. We also performed the D analysis, demonstrate that IFNA2 expression was increased in classical monocyte of COVID-19. Taken together, risk area might contribute to severe COVID-19 by increasing expression of IFNA2 in classical monocyte, highlighting the importance of single-cell-based context-specific EQT analysis to elucidate the pathogenesis of COVID-19. It summarizes. This study was co-supervised by Professor Yukinori Okara and Professor Atsushi Kumanogo. I really appreciate all the members of the team. Thank you.
Thank you so much.